Yes, thank you very much for your kind introduction, and thank you very much for the opportunity to present our data. Picosecond lasers use the mechanism of LIOP, which stands for laser-induced optical breakdown. The formation of LIOP relies on the generation of initial seed electrons released from laser-heated chromophores like melanin. A cascade occurs that ultimately results in a very localized ball of hot plasma, which then destroys local tissue with little or no collateral damage. This creates microscopic lesions in the epidermis characterized by vacuoles, and finally stimulates a healing response and consequently skin remodeling. Former in vivo studies have already shown that in addition to the laser settings, the content of melanin in the skin influences the formation of LIOP. That means that the greater the laser radiance or melanin concentration of the skin, the more likely the laser pulse will initiate LIOP formation. So for this reason, we developed new 3D skin models containing melanocytes. As you can see here from selected references, our lab has an extensive expertise in the development of standardized 3D skin models to study the molecular effects of lasers. Such in vitro models are a pretty good alternative to in vivo experiments. So after seeding fibroblasts in a collagen matrix to establish the dermal equivalent, we seeded a mixture of keratinocytes and melanocytes on top of this dermal equivalent. And then, after lifting the models to the air-liquid interface, we got a fully differentiated 3D skin model. I admit it's hard to see in this picture, but actually our models produced melanin and showed a pigmentation. And then we irradiated our models with a picosecond laser. And here we followed two approaches. A series of models were irradiated with a 1064 picosecond laser using these settings to investigate the effects of LIOP in our models. In a second approach, we additionally post-treated our models with a dexpanthenol-containing ointment to investigate the effects of this ointment on the wound healing response after laser treatment. These are our histological data immediately after irradiation of our models. Indeed, we found a microscopic lesion pattern in the epidermal part of our models. The morphology of these vacuoles match those that were reported in previous in vivo studies. After 24 hours, we found wound healing response of these vacuole-like injuries in our models. Interestingly, the wound healing progress was further accelerated by the post-treatment with the dexpanthenol-containing ointment, where we found an almost restored epidermal part. Next, we performed an NGS analysis. And here we detected a gene expression pattern reflecting the LIOP-induced stimulation on skin repair mechanisms and skin remodeling. For example, we found a trend of upregulation towards matrix metalloproteinases and inhibitors of collagens and also of heat shock proteins. Interestingly, some of these effects were even enhanced by the post-treatment with the dexpanthenol-containing ointment. And we also found an upregulation of cytokines, chemokines, and antimicrobial peptides, also indicating the LIOP-induced stimulation on the wound healing response and the skin remodeling. We also investigated the effects of LIOP on the protein level and performed immunofluorescence staining with some of the markers that we found in the NGS analysis. These data, for example, shows the expression pattern of MMP9, which is a known regulator of the wound healing response. And here we detected a trend towards an upregulation of MMP9 after picosecond laser radiation, which was significantly enhanced by the post-treatment with the dexpanthenol-containing ointment. With that, I want to conclude. We present the first in vitro study investigating LIOP effects after 1064 picosecond laser radiation using a new 3D skin model containing melanocytes. The picosecond laser treatment causes the formation of LIOP in our models, and it forces a gene expression pattern at the molecular level that stimulates the skin's repair mechanisms and skin remodeling. We found that the positive effects of LIOP could be supported and accelerated by a post-treatment with a dexpanthenol-containing ointment. It means that the vacuoles close more rapidly, but it doesn't interfere the effects of LIOP. Taken together, our data contribute to a better understanding of the biological effects of LIOP and help to optimize its application and its combination with other topical treatments. Thank you for your attention.